Hi everyone, welcome back to the introductory series for the Obesity Machine Learning competition. Today we'll be talking about the data. This is the third and final lecture. And specifically, we'll be talking about the experiment harnessing the technologies that we spoke about in the second lecture was performed to generate the data. And then we'll discuss how the data actually looks, like the data object that you'll be working with. And finally, we'll talk about the three crunches, the tasks that you'll be performing. As I mentioned, we'll break this up into three parts, going over, first of all, the Perturb-seq experiment on adipocyte cells to generate the data, then the data objects that you'll be working with, and finally, the three crunches, or the three challenges. Okay, so just a quick recap. Hopefully by now you're familiar with this scheme and the different adipocyte cells that we see in adipose tissue, some that are associated with differentiation and others that are associated with specific flavors of metabolic activity. We also discussed how these are therapeutically relevant because we know that different proportions that deviate from the normal of these different cell populations are actually linked to obesity. And finally, we spoke about the gene expression programs which govern each of these different cell populations. And we also spoke about the specific functions that individual genes within these gene expression programs have. So for example, we spoke about the capacity of the pre-adipocytes to proliferate in the presence or when PDGFRA is active, and also the ability of the adipocytes to signal to other tissues when AdipoQ is active. The issue, however, was that while we know information for some of these genes in the gene expression programs and some of their functions, we'd like a more systematic whole transcriptome view of what's going on. And so on the experimental side, we learned about this technology called Perturb-seq, which allows us to both delete individual genes in the cells and then measure the gene expression profiles in those same cells. And so one aspect of this is this gene editing technology called CRISPR-Cas9, the Cas9 nuclease, which is directed to individual genes in individual cells and inserts these double-stranded DNA breaks, breaking the function of the gene. And the second component is combining this perturbation with single-cell RNA sequencing. So this is the schematic of individual cells being isolated in these droplets, barcoded for then downstream counting of the individual RNA molecules, i.e. the expression levels of every gene. And so Perturb-seq is really the combination of these two technologies, allowing us to get the transcriptomes with the individual RNA molecules in the different cells, as well as the identity of the gene that was knocked out in that cell, in this case denoted by A or B. And so combining the biology and the technology, what's the experiment that was actually performed? So first of all, this was performed by three expert scientists at the Broad Institute, experts on adipocyte biology. And what the team did was to take pre-adipocytes and differentiate them in the lab in a dish. So this is very exciting already because it means that the kinds of gene expression programs that we've been talking about that we know exist in tissue in people, like the differentiation programs and the metabolic programs, this means that we can actually study that now in the lab in a dish. And so this differentiation protocol lasts 14 days. And so you can imagine that roughly speaking, initially we have a population of just pre-adipocytes. And after these 14 days, we may still have some pre-adipocytes, shown here in blue, but we'll also have generated this more differentiated population of adipocytes, shown in pink or red. And so this is step one. And what Tiago and the team did was to combine this with perturbations. So knocking out, as we've spoken about, individual genes in these cells at day 0, and then proceeding with the differentiation protocol. And then finally, at day 14, performing the Perturb-seq experiment to both get the transcriptomes of the resulting cells at this day 14, but also know whether which gene was knocked out in each of these cells. So shown here is just an example with one gene, let's say the red gene, but in reality, they tried many different perturbations in a pooled format, and so every cell would receive a different gene. The second experiment, which is the data set for Crunch 2, the first experiment was the data set for Crunch 1. And so in the second experiment, the setup is very, very similar. The only difference is that now, rather than single gene knockouts, what the team did was actually knock out 2 different genes in every cell. And so again, this is reflecting the perturbation and the actual double-stranded break that happens in the cells. And then subsequently, again, the cells were profiled by Perturb-seq, and those double perturbation identities, those double gene knockout identities, retrieved. So just to comment on the double gene perturbation screen, something that is worth considering when you build your models. Here is a scheme of the hypothetical perturbation effect of knocking out gene 1. So let's say it has a perturbation effect of 1. And then the perturbation effect on the cell of knocking out gene 2, which we'll say also has a perturbation effect of 1. Now, one kind of naive model might expect that the combination of these 2 knockouts, so knocking out both gene 1 and gene 2 in the same cell, would lead you to a perturbation effect of 2. This is what I call the additive expectation. But in reality, we see all sorts of interactions between genes that deviate from this expectation. So here are 2 extreme examples that I'm showing. One of what we call a synergistic relationship between gene 1 and gene 2, where actually the combined effect of knocking both of these genes out is far greater than the expectation based on just summing their individual perturbation effects. And conversely, you can also have antagonistic relationships where the combined effect is less than you would expect based on the sum of gene 1 and gene 2 effects alone. Okay, and so the output of both of these data sets in the single gene knockout and in the double gene knockout Perturb-seq of adipocyte cells, this is the output that you'll get. So on the left here, we have the single-cell RNA-seq expression matrix with every row corresponding to a different gene in the genome, and every column corresponding to a different adipocyte cell that was profiled at day 14 after the differentiation treatment. And every value inside the matrix corresponds to the RNA counts for that gene in that cell, or the as we refer to it more generally, the gene expression level. So that's one part. And then the second part is this perturbation identity matrix, where we'll be able to say for a given gene expression profile in a given cell, whether that same cell received a perturbation or not, and if it did, what perturbation it received, i.e. what gene was knocked out, or perhaps for cell 3, what pair of genes was knocked out. And then finally, it's important to note that there are also in the data set some cells which won't have had any gene knocked out. We call these the non-targeting controls. In the data set, they're referred to as NC. These are an important and revealing population because we can compare the effects of the perturbations from, for example, in cell 1, the effect of MLX-IPL, to this wild-type population that didn't have any gene knocked out at all. Okay, so just to review on the experiments, on the data sets generated by these experiments. So first of all, in terms of the number of genes knocked out, in the first data set, the data set for Crunch 1, there are 166 single gene knockouts, and we'll hold out 10 to 15% of these for evaluation. And then in the second data set, there are 105 combinations of knockouts, so 105 double perturbations. And again, we'll hold out 10 to 15% of this for evaluation. And so how were those genes selected? For this, we consulted experts in the field, adipocyte experts, and they chose genes that they believe could be relevant to obesity. And so this is the set of genes that we focused on. And finally, as I mentioned, yes, there are negative controls. These are cells that did not receive any genetic perturbation, and we call these the non-targeting controls, and you'll be able to identify them in the data sets that you receive because they'll have this NC annotation rather than the name of a gene under the perturbation column.